Good morning, everyone. Welcome to another lecture of the series of Basics of Virology. So today, what we'll be discussing. So today we'll be focusing on a very important group of viruses, which are very unique in terms of our genetic engineering tool, in terms of the usage of a virus to go for genetic manipulation, and it defines us the basic understanding or serves as a model for basic understanding of a virus. The term lytic and lysogenic that we understand as a student and that we teach as a mentor is based on these particular viruses. And yes, today we'll be talking about bacteriophages, and among them we'll be specifically talking about the lambda bacteriophage, which I think is one of the best viruses studied till date. The complete genome, the complete regulation, each and every aspect of the genome has been elaborately elaborated very beautifully by different mechanisms, and based on that understanding we use it as our vector in recombinant DNA technology also. So in this particular lecture, what we will be focusing on, whether we'll be touching the biomes of recombinant DNA technology, no, we are talking about basics of virology. So we will first understand what are bacteriophages in the layman terms, so that the students who have started this particular basics of virology course, it's not an advanced part. Yes, advanced part will be taken care of later on, but today we'll be focusing on very basic part and try to develop the basic concepts and try to have a clear cut understanding that what is bacteriophage, how it replicates inside the bacteria, what experiments were done to identify them, and then we will be talking about their genome, how they operate between lytic and lysogenic cycles. So by the end of the lecture, I sincerely hope that you will be able to completely understand the lambda genome regulation of lytic and lysogenic cycle, which is very important when we're talking about in terms of competitions also. Many questions comes regularly in competitive exams based on this regulation of lambda viruses. So let us not waste any further time. Let us begin. So see, what are bacteriophages first of all? See, if you bifurcate the word bacteria, that means the host is always bacteria for these viruses. The host is always bacteria for these viruses. And when we are talking about the phages mean to this derived from phagene means to devour. So the viruses which are infecting bacteria are called bacteriophages, and there are a number of viruses that infect bacteria. We are not talking about only T4 phage or the lambda bacteriophage. No, there are plentiful viruses which are infecting these bacteria, and they are named based on the most of them are named based on their host itself. So what they do, what exactly they do, they are very simple viruses. They will infect the bacteria, they will inject their genome inside the bacteria, and it will utilize the complete prokaryotic bacteria machinery in order to replicate its genomic content as well as to synthesize the protein which is required for the capsid formation and the packaging. And then when the things are done, when it has completely used up the bacteria, then there is no more requirement of the cell. It will burst it and it will come out. On the other hand, if the mood changes for this virus, and we'll determine how that mood changes. So if the mood changes, what it does, it injects the genome, and that genome will be integrated inside the bacterial genome. So it will remain as dormant state, and just like that we have read about the HIV part, just like the HIV part that we read, that we discussed a few days back. So what happens? It remains in the dormant state, and that particular state is called as prophage, and there will be a certain stimulus in order to activate it. In case of prophage, that particular mode of cycle is called as lysogenic cycle. Then it gets stimulated. It again follows to lytic cycle, utilizes the host machinery, increases its number, and then lyses the cell, and then multiple particles are again released. So that is how one should have a pre-notion before going in depth to the bacteriophages, how things are going. And then when we're talking about the genome part, so the bacteriophage family that we are talking about, the bacteriophage umbrella contains many viruses, and they have single-strand RNA. We have single-strand RNA bacteriophages. We have double-strand DNA bacteriophages, single-strand DNA, double-strand RNA. So we have all the groupings that are required under a single umbrella of bacteriophage. Under a single umbrella of bacteriophage. So how this bacteriophage was discovered? See, the first observation that a virus can attack a bacteria that was observed by Todd and Hurley in 1915 and 1917. So you see, we are talking about 1915 and 1917. Even when the facilities were limited, the resources were limited at that point of time. Simple concept gave rise to a conclusion that viruses can infect bacteria. What they did in the lab. See, what they observed is that when you take a bacterial broth, on that bacteria specifically intestinal bacterial broth, because they are working on the intestinal bacteria. Now, if you take a culture filtrate, if you take a filtrate which you are obtained from a sewage sample, what you do, you go for the filtration of that sample, and you will get a filtrate. You will get a filtrate. Now, if you add that filtrate in a fresh intestinal growing bacterial broth, which is in log phase, then what you observe? You observe that after incubation, the complete intestinal bacterial broth, which contains the intestinal bacteria, the lysis of the bacterial cell wall occurred. So, what they thought? What they thought? They thought that something that came after filtration, that was retained in the filtrate, is responsible for causing this particular death or lysis of the bacterial cell. And then at that time, they called it as poison, the filtrable poison. And if you talk about the dictionary meaning of the Latin language, then poison's Latin word is virus itself. Poison Latin word is virus itself. So you see, bacteriophages, when we are talking about, they carry the genetic information that is required for their own replication and increasing the number. The information that is required for regulating the life cycle, as well as the protein code that is required. The complete information is coded in their genome. So when they affect the host cell, when they affect the whole cell, their primary work is to replicate their own nucleic acid, first of all hijacking the machinery, and then also produce its complete protective protein code. So that is how these particular bacteriophages work, and this is the classical pictorial representation of bacteriophages that we are always familiar. And this is perhaps my favorite diagram also that we used to make. It's a very peculiar diagram that it contains a double-stranded DNA. There is a protein coat which is called as capsid. There will be a neck region, and then there are tail region, and it is attaches to the surface to the cell wall of bacteria. So it's a diverse group actually. And when we are talking about its general structure, then the general structure is made up of head, which is a capsid, icosahedral symmetry, and we have a neck which is of helical sheath. It's a kind of a helical structure which has a cavity inside, and there's a tail at the base with the tail pin, or the hexagonal base plate. There is a protrusion that is coming out called as tail. Now the attachment is the responsibility of these tail molecules, and when the attachment is there, it's the helical sheath which gets compressed, and the DNA which is present inside the head is injected inside the bacteria. Injected inside the bacteria. So if you look at the bottom, it's a kind of a punching machine. It's a kind of a nail pincher, where the head will be compressed, the neck region will be compressed, and due to that compression, the DNA is pushed inside the bacteria via this helical form, and the base plate, which is a hexagonal plate ensures that there is a tight attachment on the cell surface. So if you talk about bacteriophage, at present there are almost 5,000 bacteriophages that we are talking about in the single umbrella of bacteriophage. The 5,000, those 5,000 bacteriophages have been studied, and they have been divided into 13 different families. So when we talk about bacteriophages, we have 13 different families, be it myoviridae or nudiviridae, or we are talking about the podoviridae. All these viruses, there are 7 viruses which are grouped under double strand DNA, which have no envelope. They are naked double strand DNA viruses. We have double strand DNA viruses which are non-naked. They contain envelope like plasma viridae and lipothrix viridae. We also have single strand DNA viruses. There are 2 families of single strand DNA viruses, ionoviridae and microviridae. Famous ionoviridae is M13, that is used in recombinant DNA technology also. Then we have a single strand RNA bacteriophage. The example is Laviviridae. And we also have double strand RNA bacteriophage that we are calling as 5,6 superscript, and that is called as Cystoviridae. So you see, we have the spectrum, but the majority of the dominance of the 7 families is done by double strand DNA. and we have only single families of single strand RNA and double strand RNA in these bacteriophages family. With taking example of Laviviridae and phi 6. So when we are talking about these families, let us talk about actually how what is the resemblance and what is the difference between these viruses. So when we are talking about the Corticoviridae, we are talking about the icosahedral capsid. It contains the capsid layer. It contains the subalcoid double strand DNA also. When we are talking about Cystoviridae, it contains linear double strand DNA, and not only linear double strand DNA. This Cystoviridae is an example which is a triploid, which is in triploid state. It contains 3 molecules of single strand DNA. So that's a unique part. That's again a unique part. Not even this, we have Rudiviridae, which is again a double strand DNA, linear double strand DNA, and the shape is kind of rods, the helical rods. Or we are talking about uh, microviridae. Now this microviridae is very unique. Why? Because it contains single strand DNA, but it is not linear, it is circular. The microviridae contains circular single strand DNA. And then we have Laviviridae. The Laviviridae follows the quasi-icosahedral capsid symmetry, and it contains only a single molecule of single strand RNA. Only a single molecule of single strand RNA. So you see, the diversity is very high, and each and every family has been thoroughly studied in terms of their structure and in terms of the genomic content. So when we establish the understanding of these bacteriophages, among the bacteriophages, the most commonly studied are two bacteriophages, the T phage family and the lambda phage. When we are talking about the T series of the phages, we are talking about the nomenclature from T1 to T7, where the T1 is the medium virus, and 3, 4, 5, 6 are the small virus, and T7 is the largest virus. So T T series family is again very important because they contain multiple promoters which are strong promoters, and those promoters are regularly used in recombinant DNA technology also. So when we are talking about bacteriophage infecting a bacterial cell, when we are talking about a bacteriophage infecting a bacterial cell, then if you talk about the broad category that how this particular cycle is running actually, so it's very simple. It's very simple. The virus will attach with the tail. After attaching, it will substantiate that attachment with a strong binding, and the strong binding is mediated by the hexagonal plate. If you see here, it is completely attached now. Now next job is to transfer this particular genomic content inside the bacteria. So what it will do, it will compress the neck region, which is a helical shape. It will compress it, and then it will inject the DNA inside the bacteria. So you see what it is doing. It is injecting the foreign piece of DNA inside the bacteria, right? And that is the concept that is exploited if you are talking about viral mediated transformation, or I should say transduction. So that is the thing that the understanding of these particular viruses have made possible different attributes in recombinant DNA technology. And then when we are talking about the phage entering a bacterial cell, the tail binds to specific receptors which are present on the bacteria. Now when we are talking about the specificity, how specific it is. See, these viruses are very specific. If I am talking about a bacteriophage which will be infecting E. coli, then that will be specific for E. coli. It will not be affecting Salmonella typhi. It will not be affecting Mycobacterium tuberculosis. It will be specific for E. coli, and not only to the species level. In fact, you will see that if a certain bacteriophage is capable of infecting a certain type, a certain strain type inside E. coli, then it is specific for that strain type. Like if the bacteriophage is growing, flourishing in E. coli B, strain B, and we take out that bacteriophage and try to infect the same species but the different strain of E. coli K, then the efficiency goes down. Then the efficiency completely goes down. It is not able to reproduce properly. They poorly reproduce. You will see around about 1 or 2 plaque forming units in this particular category. But if you go for multiple passaging with the same E. coli K, then after multiple passaging, you will be seeing that yes, now this particular bacteriophage is able to utilize this particular strain as a host. So what happened? Due to the multiple passaging and the positive selection, the complete genome got adapted in a way that now it recognizes E. coli K as a host. And then again, if you use this These bacteriophages that were earlier growing in E. coli B now they have adapted for E. coli K to reinfect E. coli B. Then again there is poor reproducibility. The reproducibility is very poor. Why? Because right now they are adapted for E. coli K and have forgot the past history, and that much high specificity is there for these particular bacteriophages, and that specificity can be attributed to one of the factors of attachment on the surface of the bacteria and the interaction between the tail and the phage receptors. So you see, when we are talking about the bacteriophages and talking about how it replicates inside the bacteria, it follows two broad categories. One is lytic, another one is lysogenic. Lytic is simply lysis of the cell. The phage will infect, it will multiply, it will grow in number, and will cause the lysis of the host cell and will release the multiple particles to infect the other cells. Lysogeny is the phase where the injected DNA will be getting integrated inside the bacterial genome, and at that particular moment the bacterial host chromosome will be holding the viral DNA and will be stable and it will be continuing in that stable state until a time is reached where the stimulation is provided, and then perhaps it can again come into lytic phase. And can perform the complete lytic cycle. So there are two modes of replication. Suppose the lambda virus is getting attached and it is injected its linear double strand DNA. Now what is the job? The linear double strand DNA will first circularize because it cannot be in the linear form. To prevent the degradation by the nucleus, it gets circularized. Now in order to circularize, you must understand, if this is to circularize, then these two ends should be complementary to each other, then only they will be able to bind. If there is no complementary, then no binding will be taking place. Now this particular complementary or the terminal regions of the single strand DNA are called as cos sites, and these cos sites can combine to give rise to circular double strand DNA. Now after forming the circular DNA, it can go either in lytic mode, or it can go in lysogenic mode. What is lysogenic mode? Now this particular circular double strand DNA will find its complementary region inside the bacterial host genome, and by recombination it will get integrated inside this particular genome, and with the help of the enzymes integrase that are coded by this particular virus also. So the expression of the gene which are present on this circular double strand DNA will dictate how this particular virus should follow, whether it should go to lysogeny or it should go to lytic. If it decides to go to lysogeny, then all the genes, all the proteins that are required to add this particular viral DNA, integrate this particular viral DNA inside the host chromosome, that will be expressing and that will perform their function. Once this particular virus has integrated in the bacterial genome, then this particular cells are called as prophage, and they will be very stable. They will be keep on replicating. There will be normal cell division, and then there will be multiple copies of bacterial cell which contains the lysogenic state. And upon induction, upon stimulation after a certain period of time, then this particular integrated part can be detached and can maintain again as a circular double strand DNA, and then it can opt to go to lytic cycle. Now what is lytic cycle? Once it is in a circular double stranded form, it decides that I will not be integrating. Now I will start my life cycle. What it does, it synthesizes proteins. What proteins? It synthesizes proteins which will cause the replication of this circular double strand DNA, as well as the head protein, the neck protein, and the tail proteins which are required for the complete packaging. Those are formed, and then the packaging starts. And once the burst size is achieved, then the cells get lysed, and then the complete bacteriophages are released. So this is how the complete bacteriophages. The bacteriophages are following its complete cycle, whether it is lysogeny or lytic part. So when we are talking about the lysogenic conversion, how that lysogenic conversion may occur, how the prophage, which has the integrated viral genome, can again go to lytic cycle. So it depends upon the co-infection part. See, The changes that stimulate this particular change from lysogenic to lytic can be many. It can be the temperature condition. It can be nutritional starvation. It can be nutritional starvation. It The most of the time these changes, what we are talking about, the changes which are induced in the phenotype of the bacteria, that is in fact not even the part of that phase cycle. What is happening? The changes are appearing on the cell membrane, the cell wall, which is present. The changes occur on that. See, that is the these are the conditions. These are the conditions where the conversion is happening. Where the conversion is happening. Let us discuss this with an example, like of classical example of Corynebacterium diphtheria. What it does? This particular Corynebacterium diphtheria produces the diphtheria toxin. When it produces the diphtheria toxin, which causes the ADP ribosylation and inhibition of translation, under normal condition, if the Corynebacterium diphtheria is present as a wild type, and if it infects, it will never be able to secrete diphtheria toxin. It secretes diphtheria toxin only when it's in the form of prophage. When it is in the form of prophage. If this particular diphtheria, Corynebacterium diphtheria, is infected by a phage beta, in that case, that phage beta will be following the lysogenic cycle, and that lysogen, or the prophage that is actually Corynebacterium diphtheria, that will be responsible for secreting the diphtheria toxin. And similar is the case of Vibrio cholerae. It never secretes cholera toxin until and unless it is infected and it is under the lysogenic control of phage CTX5. Phage CTX5. So you have to understand that these bacteria have the viral genome inside them, and that's why they are secreting the toxins. That's why they are secreting the toxins like Clostridium botulism, which is causing botulism, or we are talking about cytotoxin. So depending upon the bacteriophages, there are different like Vibrio cholerae, which is causing cholera. It is getting infected by the CTX phage, and that's why it is secreting the cholera toxin. Whereas when we are talking about, let's say, E. coli, infected. It is getting infected by the lambda phage, and it secretes cytosol-like toxin. Or when we are talking about the Streptococcus pyogenes, it secretes erythrogenic toxin for the T12, which is responsible for causing the scarlet fever. So this is how these particular viruses go about opting for whether the lytic mode or the lysogenic mode. So what controls this? What exactly controls this? So when we look at its genome, and when we talk about this particular aspect, that in this the complete circular double stranded DNA, what we're talking about, how this regulation of lytic and lysogenic occur. Let us see. See, before that, we should understand how this genome is distributed. This is a circular double-strand DNA of lambda phage, and when we are talking about, we have shown here the leftward transcription, the genes where the promoter is there, and that expression of that promoter will result in transcription or formation of mRNA from left-handwise or anti-clockwise. And then there are certain promoters which are going for right or clockwise, rightward transcription. So when we distribute them, the rightward transcription is required for DNA replication, where the genes of C2, CRO, OPQ are required, and also the genes which are required for head, tail, and lysis part are present on the right hand side. So when the right hand side of that lambda genome is getting active, it makes up all the DNA replication proteins that are required. It makes up all the packaging protein that is required. It makes up the factors which causes the lysis of the host cell. So what it is doing, it is actually performing lytic cycle. So if excuse me, if the genome is getting expressed in the right hand part or the clockwise direction, then it will always go for lytic. And if it is going for left hand direction, where the genes of zis, int, and at, zis is for excision, int for integration, at for prophage ends, if these genes are getting expressed along with C3 and C1, which is actually a repressor protein, these portions, the left hand panels, are required for lysogen. So after infection, if the promoter of left hand side, the PL, is getting active, then of course it will be going for lysogeny. And if PR, that is right hand side promoter, is getting active, then it will go for lytic mode. And these promoters, how they will be getting activated? which proteins are required for their repression or their activation, and that is the job that we will be discussing in terms of in terms of regulation of your lytic and lysogenic cycle. So in terms of regulation, when we're talking about that which part, which protein gives lytic, which protein gives lysogeny, then that particular regulation is called as molecular switch of lambda bacteriophage. And see here, see here, the yellow portion, we have a single gene here, that is C1 gene. If this C1 gene gets activated, then the complete lambda phage will be going for lysogeny. And if this particular CRO, that is CRO protein, if CRO protein gets activated, it will be going for lytic cycle. So it should be very easy for you to remember. CRO is lysis, chrolysis, and C1 is for lysogeny. C1 is for lysogeny. So these two proteins are the marker proteins which are actually at the center of that molecular switch. At the center of that molecular switch. So if you zoom that particular part, it appears in such a manner that we have a C1 gene whose promoter is there, the PC1 promoter, promoter of C1, which is going on leftward direction, and we have PL, that is a promoter which will go for left hand side and it will cause the expression of the other proteins. What other proteins? The C3 and this, int and at. So PR is the promoter which will be going towards clockwise direction or right hand side, and it will cause the expression of CRO protein, and it will cause the expression of CRO protein. So when we are talking about C1, when we are talking about C1, what is C1? C1 is actually a repressor protein. C1 is actually a lambda repressor gene. So what is happening when we are talking about lytic cycle? How the lytic cycle is getting ahead? For lytic cycle to go ahead, the piece promoter of C1 needs to get inhibited so that there is no expression of C1. If there is no expression of C1, then there will be no lysogeny. Only PR will be activated and CRO proteins will be released, and these CRO proteins will be responsible for forming lytic cycle. So what is the condition of lytic part? The condition of lytic part is promoter of C1 should be turned off, whereas the promoter of right hand side PR should be turned on, as well as PL. So when you're talking about lysogeny, in terms of lysogeny, only and only P promoter of C1 should be activated, whereas the other two promoters should be turned off. And only C1 protein should be expressed, which will be a repressor protein, which will repress the activity or the lytic activity of this lambda phage. So what exactly is causing these turning these promoters on and off? What exactly is turning these promoters on and off? So if you look at this particular region, which we are talking about, the C1 and the CRO, in between these two genes there are two promoters, promoter C1 and promoter R, that we clearly understood. But along with these two promoters, there are three DNA motifs which are called as operator regions. Now these operator regions are arranged in such a manner that OR1, that is operator 1, is close to CRO protein, and it is overlapping with the PR. OR2 is again overlapping with the small portion of PR, whereas OR3, the operator 3, is completely overlapping with promoter C1. Now what happens next? See, because of the overlapping region, this is the arrangement that you finally see when you merge these two. This is the arrangement that you see from C1 to CRO. Now what is happening here? Now suppose lytic cycle is here and PR gets activated, it will cause the formation of CRO protein. Now this CRO protein as a dimer will go and bind to operator 3, OR3. Now OR3 is overlapping with PC1, so it binds to the operator region, thereby it prevents the RNA polymerase binding to the promoter C1, and that is how the expression of CRO protein inhibits the expression of C1, thereby it inhibits the expression of lysogeny. And then only the complete lambda phage follows the lytic cycle and it flourishes. So what is happening? The expression of CRO protein, the expression of CRO gene to form CRO protein is actually responsible for suppressing the lysogenic cycle by inhibiting the binding of RNA polymerase to promoter C1. And how it inhibits? in the dimeric form and bind to the operator region, that is operator 3 OR3, which is overlapping with the PC1, the C1 promoter. Whereas when we are talking about that lambda phage will go for lysogeny, then what happens in when we are talking about lysogeny, then this PC1 is getting activated and it goes for the expression of C1. Now this C1 forms a C1 protein called as repressor protein, and tetrameric form of that repressor proteins comes and binds to OR2 and OR1. The OR1 which is overlapping with PR and OR2 which is overlapping with PR. So it binds to both the operators. If you talk about the C1 protein, it's a kind of, the structure is kind of a helix turn and helix. What it does, it prevents the binding of RNA polymerase to the promoter region, and thus there is no expression of CRO protein. If there is no expression of CRO protein, there is no inhibition of C1 gene, and if there is no inhibition of C1 gene, then of course the cell will prefer to go to lysogenic path. Now when we are talking about this particular aspect, that yes, C1 protein is going about, and that C1 protein is in binding to OR1 and OR2, thereby it prevents the binding of RNA polymerase to the promoter region, and that's how it shut down the CRO expression. Now the interesting thing is, once a tetrameric molecule is going and binding to the operator, then there is a cooperative binding. It will further potentiate the binding of other C1 proteins to all the operator regions. Now what is happening? Slowly, this cooperative binding leads to the occupancy of OR3 also. Thereby, it inhibits the binding of RNA polymerase to its own promoter. Now, what it indicates? It indicates that the lambda phage has now suppressed, the C1 protein has now suppressed the complete PR and PC1. So no more C1 is required. So the complete infected double-strand DNA will now be going for lysogeny. So that is the reason that after initial suppression of lytic path, then after some time, even the expression of C1 protein or the repressor protein lowers down or diminishes, because the initial C1 proteins which were formed occupy the promoter region, which is responsible for the expression of both lytic and lysogenic regulator switches. So that is how the complete regulation is going about. That is how the virus, this particular lambda bacteriophage, performs its cycle, whether it is lytic and lysogenic, and that is how it takes the decision based on the CRO and C1 protein, whether it should go for lytic or it should go for lysogenic. So it's a very beautiful part that we understood, that this is how the regulation is going on. Now the question comes, a beautiful question. When we're talking about the lecture, then you should always be focusing in terms of broader picture when we're talking about the competitive exams. What will happen? The question, please listen carefully. What will happen if there is a mutation in PC1? There is a mutation in PC1. The promoter of C1 is mutated in such a way that it is not able to bind to RNA polymerase. In that case, there is no question of lysogeny. The lambda phage will always follow lytic cycle. If there is a mutation in promoter R, PR, then there is no point of lytic cycle. The double-strand DNA will always go, the viral genome will always go for lysogeny. And that is the way we manipulate this particular lambda genome inside our lab to inhibit its lytic cycle. You can inhibit the promoter region. Even if you can cleave off the genes which are actually required for lytic cycle, because you don't want your lambda genome which is being used as a vector to go for lytic mode. You want it to get integrated. If you want it to get integrated, then you can remove that lytic genes from this particular genome, and instead you can ligate or you can insert any foreign gene of interest to go for a vector DNA hybrid. Next question, which is very interesting one. If there is a mutation in operator 3, OR3, then what will happen? If there is a mutation in OR3, then the crow protein will not be able to bind to OR3 and will not be able to inhibit the C1 protein. On the other hand, if OR3 is mutated, that means the PC1 has the mutation. The PC1 has the mutation. So will it bind to RNA polymerase? Yes, if the mutation permits and there is a good binding of RNA polymerase, it can still cause the expression of C1 protein. It can still cause the expression of C1 protein. But you will see that there is no suppression in, after some time, there is no suppression as we have observed in cooperative binding of C1 protein. You will always see C1 protein activated. Why? Because it never inhibits the PC1 because of its mutated part, it never binds. So these are certain questions that one should be preparing in terms of mutational based studies. What will happen if this is mutated? What will happen if that gene is mutated? Now, just to give, although see these bacteriophages are used in multiple biomes, whether we are talking about the medicines or whether we are talking about the R&D sector. Just to give you an example, a term of phage therapy that was introduced in 1940s in Soviet Union when the army was focusing more on treating their soldiers with having the bacterial infections, because rather than getting killed by a bullet, they were more affected by bacterial infections, and the prolonged war was causing havoc in the base camp, army base camps. So what is happening? What they designed a phage therapy. They came to know that yes, these phages can lyse the bacteria. So why not to use these phages to lyse the bacteria, which is actually the pathogen on humans? So what they did, they performed widespread clinical trials. They kept on treating the soldiers in the Red Army. However, later on it was abandoned because of multiple reasons, because of multiple side effects, which we now understand that those side effects were based on viral infection. These were mainly allergies which were not handled and lead to the havoc, lead to the different side effects or aggressive side effects also. So it was at that time on 1940s, it was the basic lack of understanding about the phage, about the immune system, about the immune evasion and the immune response, which led to stopping the use of bacteriophages in medicine. But then when we had better instrumentation, when we had the better concept orientation, and when we had understood major part of these viruses or these phages, then again this particular research was potentiated, and in 2006 the FDA, Food and Drug Administration, approved in fact the use of LMP102 as food additive to kill monocytogens, Listeria monocytogens, or uh, after treating with LMP102, they tagged it as ready to eat poultry or meat products. Those meat products are still being treated with this particular LMP102 in order to kill any kind of presence of Listeria monocytogens. Later on that year itself, in 2006, October, later that year, another approval for LMP102 was given. It was given again. It was given to kill Listeria monocytogens only, but the approval was given to use this LMP. Earlier, it was given for meat products and poultry. This time, it was given for cheese. It was given for cheese, that this particular pathogen also deteriorates the cheese, so it is allowed to use this particular LMP102 in cheese also to increase the shelf life. And they clearly identified that this particular LMP102 is give is grass. What is grass? Generally recognized as safe. So this is how the complete story of bacteriophage culminates. And for your further reading, I have given the link of two very beautiful and very in-depth insights on bacteriophages, which will again clear your understanding on this topic. So let us talk about the MCQs. Now these MCQs are of utmost importance. These MCQs actually shows how much we have inculcated. So let us take those examples. Yeah, it is very important to study lambda biology, as lambda phages are used for cloning purposes, as I told you in genetic engineering. Which of the statement is true for lambda phage? It's an example of temperate phage. What are temperate phage? The temperate is another synonym of prophage. The fate of the phage is decided before it infects the cell. Before infection, is there any life cycle which it performs? The lysis fate is that where the phage inserts into the bacterial genome, and the replication goes on. Or the lysogeny fate is that where the phage infects the cell and lysis is carried out. So this is how you have to identify which statement is true. Whether these two are contradictory or which one is true, it's up to you. Once you go through the lecture, you will be able to know. The immediate early transcript direct synthesis of which gene? First of all, the immediate genes which are transcribed. What are those genes? Is it N, CRO, PR, or PL genes? Or which one of them is correct about lambda phage? It is a single strand genome. It is circular double strand genome. The ends are cause sites. The ends are created by cleavage at cause sites, or it's pure glucose. So when we are talking about these options, you should first go through the concept, and then only you should be able to answer these particular MCQs, like which of them is again correct statement for C2 gene and C3 function. So in further reading, you may further understand what is the role of C2 function. We have clearly understood what is GROW, what is C1, but we have not touched C2 and C3. So you should understand what is the critical role of those C2 and C3 also. So with this, we should culminate our learning of this particular topic, that is bacteriophages. We today understood clearly that how this particular bacteriophage infects, whether it is lytic mode or lysogenic mode, and at what point the molecular switch, how that molecular switch is regulating the lytic part or the lysogenic part, what is the role of CRO in lytic and suppressing lysogeny, or what is the role of C1 in suppressing lytic and potentiating the lysogenic mode. So this is all about this is all about bacteriophages from my side today. So I hope the lecture was fruitful to you, and we will again meet with another topic in the series of basics of virology. Till then, take care, please stay safe, and wish you a very, very happy learning. Thank you so much.